Hey guys, so this video is over photosynthesis, just the very first set of reactions and just a little bit of background information. We're going to go over some things that you're already familiar with from your previous biology classes. Just for the sake of you keeping abreast with the order here, you should have already looked at the ATP video before you view this one. So if you haven't looked at the ATP video, make sure you go back and check that one out. Alright, so we're going to go ahead and start. Okay, so let's talk a little bit about who does photosynthesis. And normally we tend to associate the act of making your own food with being a plant, but as you are aware, that's not always entirely true. There are some organisms that are not actual plants, but are capable of doing photosynthesis. But they all have the same needs for the most part. And those needs are, they have to be able to collect energy in the form of light, and then they have to be able to take that energy and transform it into the chemical version of energy. So again, it goes back to that whole law of conservation of energy where you can't create it, you can't destroy it, you just change the form that it's in. Once the energy is harnessed, you have to be able to store it. You have to put it in some kind of stable form where it can be moved around the plant or stored until it's needed for later use. They need to be able to build atoms from their environment, and they're going to be utilizing some very common biological elements to do so, things like carbon and hydrogen and oxygen and nitrogen, and those elements that we talked about previous to this video. Through a combination of getting energy and then being able to synthesize things, they need to make organic molecules. So they're building their own molecules for use. And those organic molecules are going to be used for growth. And of course, whenever we talk about organic molecules or biomolecules, we're talking about carbs, proteins, lipids, and nucleic acids. Okay, so once you are alive, you're going to need a constant supply of energy. You're constantly utilizing energy for your life. So some animals get their energy by eating other things, like heterotrophs, for example. Any animal, for the most part, is going to be a heterotroph. You are eating something else for food. You are getting your food source from another living thing. And energy for these organisms are made through the process of respiration, which we will talk about a little bit later on. Now, you have to remember that when we talk about energy and we talk about food, what we're really talking about are those organic molecules. Another name that we could use for heterotrophs would be the words consumer, because you are consuming something else. On the flip side, if you're not a heterotroph, it means you are an autotroph, which means that you can make your own energy yourself. You're producing your own food. And you're doing that by converting energy from the sun into organic molecules that you will use to build things from. So you make your energy and you synthesize sugars through the process of photosynthesis. You are an autotroph, or you are a producer, because you're producing your own food. So how do we connect them? Well, let's think about it. Heterotrophs utilize other things for food, but they're still making energy, they're still making organic molecules, but they're doing so by ingesting organic molecules that already exist. Whereas autotrophs are making energy and organic molecules, but they're doing so from light energy. They're harvesting sunlight for this. So if you look at the reactions, they kind of look the same. Heterotrophs seem to be doing the opposite of what autotrophs are doing, and that's kind of true. Autotrophs are doing photosynthesis, whereas heterotrophs are doing cellular respiration. And we'll talk about exactly what that is a little bit later on. What I want you to pay attention to is, where is the ATP in all of this? When a heterotroph does respiration, it is making ATP at the end, but notice you don't see ATP here on this equation, that represents photosynthesis. So we'll talk about where that ATP comes in a little bit. Okay, so if you are an autotroph, it means that you're making your own food. The majority of the autotrophs that we deal with are going to be plants. So let's go over the basic structure of a plant real quick to help us remember what all the parts and pieces are for. Now, a plant really has evolved its structure in order to be good at doing photosynthesis. The first step in photosynthesis is getting the necessary raw materials. The first major raw material is going to come in the form of sunlight. So that's the job of our leaves. Our leaves are going to act like these big solar panels to collect as much sunlight as possible. You also need carbon dioxide as a reactant in this photosynthesis equation, and that's also going to come from a section of the leaves called the stomates or the stomata. That's where gas exchange happens. Just to remind you, because we've already done plants, your stomata is just going to be that little opening in your leaf that opens or closes and allows air and other things to come in and out. We're going to need water. Water is going to come from the soil, so our roots are going to be in charge of harnessing and pulling water up to our leaves where photosynthesis is going to take place. And we're also going to need some micronutrients like nitrogen and potassium and sulfur. And again, that's going to be dissolved within the water in the soil, so the root system is still in charge of pulling those micronutrients up into the leaves. So let's go over some quick pictures over the structure of the leaf. So if you looked at it, this is a cross section of the leaf. So we've taken the leaf, we've cut it in half, and we're looking on the edge that we've cut. These cells right here, which make up our mesophyll, that's where the majority of photosynthesis is going to take place. Notice that this big circular component right here to our leaf, that's going to be where our xylem and our phloem are residing, so that's where our water is coming from. And then where the arrows are pointing out and in, that represents where our stomata are, so that's where gas exchange is going to take place. Okay, so along with having these leaves and these roots, we also need an organelle in which photosynthesis is going to happen. That organelle is called a chloroplast. So, brief overview of the chloroplast. It is a double membrane, so it has two membranes. The outer membrane is just a surrounding membrane, and then just inside of that outer membrane is an inner membrane. So they're pretty much one on top of each other. See if I can get my laser back. I keep moving the slide. Hold on. There we go. Okay, so inside of the actual chloroplast is a fluid-like interior or fluid-filled interior, and that is called the stroma. So all the liquid parts of the chloroplast is called the stroma. And then we have these specialized membranes or sacs or stacks. So sacs and stacks. Try saying that 2 times quickly. Which are called the thylakoids and the grana respectively. So let me see if I can explain the difference for you. A thylakoid, also pronounced as a thylakoid, so whichever you're comfortable with, would be an individual green disc on my picture. So like right here, that single green disc would be a thylakoid. This would be another thylakoid. A granum would be one stack of thylakoids. So like here, if you follow my laser, it looks like there are about 10 of them stacked together. That would be a granum. Grana would be several stacks of thylakoids. So think of it as stacks of pancakes. A single pancake would be your thylakoid. A short stack, where you get like 4 pancakes one on top of each other, that would be a granum. And if 4 of us went to Denny's or, one of those breakfast places and we all got 4 stacks of pancakes, so there are 4 sets of 4 pancakes, then we would have grana. So hopefully that made it clear. So inside of these membranes, inside of these thylakoid membranes, that's where you're going to find your chlorophyll molecules, and it's also where you're going to find something called the electron transport chain, and a very special enzyme named ATP synthase, which makes ATP, just like the name suggests. All right, so what is exactly photosynthesis? Well, photosynthesis is a process, or actually it's a 2-part process that is divided into what we call the light-dependent reactions and the light-independent reactions. So your light-dependent reactions need light. That's where your energy conversion reactions are going to take place. This is where we are taking solar energy and we are going to convert it into chemical energy. Okay, so solar energy goes in, and if you follow my little diagram here, we'll also make ATP, which is our chemical energy, and then we'll make NADPH, which we'll talk about what this is in a little bit. So that is step 1 of photosynthesis. Step 2 of photosynthesis is called the light-independent reactions, not the dark reactions, because it does not happen at night only. It can happen at any point in time during the day. It's also known as the Calvin cycle because it was named after the person that discovered it. So the Calvin cycle, or the light-independent reactions, is where we take that chemical energy that we've made and that NADPH and we use it to make sugar. We use it to make our molecule for food. All right, so let's step through the light reactions in a fair amount of detail. So everything pretty much works based on this electron transport chain. So just like the name suggests, we're transporting electrons. We're moving around electrons. And we are going to see this very same electron transport chain in cellular respiration when we get to it. It works because there are a series of proteins that are found on the membranes of these thylakoids. And it works because, along with the proteins, we have molecules called electron acceptors. In this case, our molecule is NADPH. Think of it as storing, change for later use. So you're storing a little bit of energy, quote unquote. Actually, you're storing electrons is really what you're doing for later use. You're going to save it up until it counts. So the way the whole process works is it's based on a proton gradient. And we're using protons in the form of hydrogen ions. And those hydrogen ions are going to build up on one side of the membrane, and then they're going to diffuse through the membrane through this ATP synthase enzyme, and they're going to make ATP. I promise you, we'll go over it again, we'll do some activities, and later on, especially when we hit respiration, this will make a whole lot more sense. Okay, so we know that we have to capture light energy, but how are we going to do it? Well, we're going to do it using pigments designed specifically for photosynthesis in the form of chlorophylls. There are two kinds of chlorophyll, chlorophyll A and chlorophyll B, and then some other pigments. And all of these pigments are found embedded in the thylakoid membrane, and they're arranged in what we call a photosystem. So a photosystem is just a collection of molecules, and they all help with the process of photosynthesis. It's their job to harness light energy. So real quick, let's just remind ourselves of the colors in the visible spectrum of light. So everyone should be familiar with Roy G. Biv, if we spell it backwards, and the different wavelengths that they are associated with. If I were you, I would write these out. So like 380 to 350 violet, and 350 to 500 indigo, and just write them out so you have an idea of where those wavelengths fall and what color they will produce. Remember, the lower the wavelength, or the shorter the wavelength, the higher the energy, and the longer the wavelength, the lower the energy. Okay, so how are we going to do this absorbing of light? We know we need to do it. We need to absorb light energy before we can make chemical energy. Well, the plant's answer to that is in the form of chlorophyll A. And it has some other pigments, but chlorophyll A is the main light collecting pigment that your plant is going to possess, or anything that's photosynthetic is going to possess. So chlorophyll A absorbs light best in the red and blue wavelengths of light, and it absorbs the least in the green section of your spectrum. So that's kind of why plants are green, because that's where they absorb color the least. The accessory pigments they absorb in different spectrums. So if you look at this graph right here, it gives you a good idea of where everything is absorbing. So you can see that your chlorophyll A is absorbing decently over here in this bluish section, and then again over here in this reddish section. Whereas chlorophyll B absorbs well in not really the indigo section, but like the true blue section, and then it absorbs again in that yellow orangish section. And then you have another kind of pigment called carotenoids. You also have another kind of pigment called xanthophyll. So there are lots of different pigments. And you can see that your carotenoids absorb well in the blue to green region, and then they don't really absorb so well anywhere else. Maybe again in like this purple region. So just so you're aware. Different ones absorb in different areas of the visible light spectrum. So how does it work? Well, I told you that the molecules, the chlorophyll molecules and the pigment molecules are arranged into photosystems, and there are two of them, and they're all found in the thylakoid membrane. Their job is to gather light. So they're light-gathering molecules. They're collections of light-gathering molecules. So there are two photosystems, photosystem 2 and photosystem 1, and the difference between the two is what kind of chlorophyll is your main pigment or your main light-gathering molecule. Photosystem 2 works with chlorophyll A, and it absorbs in the 680 nanometer wavelength or red light, and photosystem 1 works with chlorophyll B, and it absorbs in the 700 nanometer wavelength of red light. So that's the difference between the two. Photosystem 2 is P680 because that's where it absorbs in the 680 nanometer wavelength of light, whereas photosystem 1 is P700 because that's where it absorbs in the 700 nanometer wavelength of light. So that's the difference between the two. All right, just to give you an idea, so if you follow my laser, this whole thing here is going to represent the membrane system of your thylakoid. And then these purple blobs, those represent your protein molecules, or your protein, sorry. And this big one here is where photosystem 2 sits, and then this one here is where photosystem 1 sits. So just to give you some perspective on where the photosystems are actually found. Don't forget, we're still talking about light-dependent reactions. So let's go through how it works. So the whole thing is based off of sunlight coming in, and sunlight is going to hit somewhere on that chloroplast, somewhere on one of those thylakoid membranes, and that's where these photosystems, these pigment molecules, start going to work. They're going to absorb the energy from this sunlight, and then they're going to pass that energy on until they get to this P680 region of your photosystem, just kind of in the middle of your photosystem. Now, in that P680 region sits two electrons, and those electrons are of low energy. They're not what we call excited electrons, so they're just kind of sitting in that main accessory molecule. Once that energy has been passed on, the electrons get excited and they change state. So they actually have energy in them, so they're moving, they're excited. Now, once those electrons get excited and they leave that main region of your photosystem, these electrons now need to be replaced. So they're going to be replaced by electrons coming in from a water molecule. So remember how water is one of the reactants of photosynthesis? This is where it ties in. You're going to lyse or split that water, and the hydrogens get cast off, and we're going to use those for the electron transport chain, and the oxygen gets released. So that's what, you know how they say how that plants breathe out oxygen, they take in carbon dioxide and release oxygen. This is where the oxygen is coming from that plants are going to release. Now let's go back to these hydrogens. Once we've removed an electron from that hydrogen, we've made it an ion or a proton. So we're going to save these hydrogens to use it in the electron transport chain, but we're going to take the two electrons that we took from these two hydrogens and put them back into the photosystem so that the process can repeat itself. It's called filling the electron vacancy. All right, so I hope that makes sense. I'm going to give you a little bit of time so that you can look at this slide, and if you need to pause, feel free to pause. Okay, so once these electrons get excited and they change state, they're going to start moving. As they move, they're going to lose some of that energy. So that's why they're going from kind of like a high state to a low state. And as they pass through, any hydrogen, any protons that we generate are going to start diffusing through, going through that ATP synthase enzyme and creating ATP. Any ATP that we make at this stage in the cycle, in the light reaction, we are going to send to the Calvin cycle to be used to make carbohydrates. So the point of your light reaction is to generate ATP for the Calvin cycle. Okay, so we're going to generate ATP for the Calvin cycle, but we're also going to generate some electron acceptor, NADPH, for the Calvin cycle as well. All right, so far we've talked about just the process that the electrons and the light's going to have on photosystem 2. So again, that light comes in, we're going to split some water molecules. Wait, let me rephrase. So the light comes in, it's going to get passed on to the reaction center of P680, where it's going to excite some electrons, and those electrons are going to change state. We need to replace these electrons that we've just excited, and we do so by splitting the water that our roots have taken up. We're going to have the oxygen leave through the stoma, and we're going to take electrons away from the 2 hydrogen molecules we have left to replace the ones that we already lost. Those hydrogens are going to get stored for later use in the electron transport chain. Now, these excited electrons are going to start getting passed on from protein to protein, and as they get passed on, this flow of electrons is going to provide energy to make ATP. Now, literally, we are changing state. We're excited right here, and then as we move through these proteins, we get less and less excited, and then we're not excited anymore. By the time we're not excited, we are now in photosystem 1. We are in the reaction center of photosystem 1. And the process is pretty much going to start all over again. We're going to have light energy come in from the sun. That light energy is going to excite the electrons that are now in the reaction center. They're going to get excited and change state. And then again, we're going to have to replace those electrons that we've lost. We always have to keep filling the electron vacancy. So once these electrons are excited, we're still going to pass them. And this time, as we pass them through the electron transport chain, we are going to store them. We're going to attach these electrons to an electron carrier known as NADPH. And NADPH is going to be also sent to the Calvin cycle to help with the making of our carbohydrates. So the way I like to think of it is, think of your NADP as your piggy bank. It's where you put all your loose change. By itself, the change doesn't really help you out. You can't really buy anything with a penny. But if you keep storing up your pennies, over time, those pennies are going to become $1, $2, $5, $10, and now you can actually buy something with them. So individually, we may not be able to do a whole lot with a single proton, but if we bank them, we can cash them in later on for lots of stuff. That's the premise of the electron transport chain for photosynthesis. So I want you to realize that in the first part, between photosystem 2 and photosystem 1, so in this section right here, we do make some ATP. But once we've excited those electrons from photosystem 1, we're not making ATP anymore. Now we're just going to add them to NADPH. We are generating NADPH. All right, so here's an overview of the whole process. So here's photosystem 2, here's photosystem 1. Our light energy comes in, we excite the electrons change state, they start moving. We need to replace the electrons we've lost, and we do so with water. So water gets split. The oxygen from water leaves through the stoma and exits the leaf, and those hydrogens get their electrons removed to replace the ones we've already lost. We hold on to the hydrogens and we start creating some ATP because we keep storing up hydrogens. Okay? The electrons that we've already excited, they started to lose some of that excitement as they move, and now they're in photosystem 1, and we repeat the process all over again. Again, sunlight comes in, the sunlight's going to excite those electrons, and they're going to move again, but this time, instead of generating more hydrogens, we are going to just add these electrons to NADP, making it NADPH. So at the end of the light reactions, we have made ATP and we have generated NADPH, and we are going to send those to the Calvin cycle so that we can make some sugar. Okay, so let's go over in note form the electron transport chain of photosynthesis. So the electron transport chain, or the ETC, uses light energy to make two things. This is what I need you to understand, guys. This stuff right here. This is the important stuff. Okay, so here we go again. The ETC is used to make two things, and it uses light energy to make these two things. The two things are, or the two products are, ATP and NADPH. This is usable energy, and this is, think of it as stored currency for later on. It does so by utilizing two photosystems. Photosystem 2 is the first photosystem that we will get to on our thylakoid membrane, and photosystem 2's job is to absorb light. Once that light has been absorbed, excited electrons are going to go from chlorophyll molecules that's in the photosystem to that middle region where they had their letters written P680, or the letters and numbers written in the pictures, and that is called the primary electron acceptor. You need to replace the electrons that were in the chlorophyll, and we're going to do that by extracting electrons from a water molecule. So those electrons are going to get passed back to chlorophyll so that when the process is ready to repeat itself, there are electrons already there and waiting. So we split water, and the oxygen combines with another oxygen that's already in the chloroplast to form oxygen gas, and oxygen gas gets released into the atmosphere and we can all breathe. So we couldn't live without plants because that's where a lot of our oxygen is coming from. Okay, we're going to skip this slide over. All right, so there are two ways that these photosystems work. The first way we're going to talk about is the way that we want it to work, the general way that it normally works. And it's called non-cyclic photophosphorylation. So you know that phosphorylation, is about moving a phosphate group. Photophosphorylation is about moving that phosphate group, but this time we're doing it with the help of photons in the form of light energy. The word non-cyclic should tell you that this is not a cycle. It's not going to repeat itself over and over again. Instead, it's going to be almost like one way in, one way out. So these light reactions, their whole purpose is to make ATP and to make NADPH. They do it in two steps utilizing photosystems 2 and photosystems 1. Photosystem 2 absorbs light in the 680 nanometer region of the visible spectrum, whereas photosystem 1 absorbs in that 700 nanometer region of the visible spectrum. Photosystem 2 is all about making ATP, like actually making ATP right then and there, whereas photosystem 1 is all about making NADPH, which is like photosynthesis change. Remember, we talked about it being like coins, and we're saving them up until they altogether have more value than they do individually. So if you look at this little picture that I have going here, this pretty much generates how the whole thing is going to work. So our photon is our energy. It comes in, and our electron gets excited, and it changes state. It literally goes upwards. It changes levels. Once it goes up, it's going to start being passed from protein to protein all the way down, and as it does that, it is making ATP because we are harnessing those hydrogens, those protons. We're creating a proton gradient. So this whole section right here would be photosystem 2. Once we get to this section, we're now in photosystem 1. So the process kind of repeats again. Again, we have more photons of light coming in. That electron's going to get really excited. It's going to change levels. And this time, instead of making ATP, we're going to start making NADPH. So we just looked at non-cyclic. Now let's look at cyclic. Cyclic is what happens if, for some reason, there isn't any NADP for the photosystem 1 to pass those electrons to. So in cyclic photophosphorylation, we are going to make a circle. We're going to take the ending product of something and kind of bring it back to the beginning and do that step all over again. So if for some reason photosystem 1 can't pass its electron to NADP, we're not going to just stop the process. That would be kind of crazy. So we're going to take that electron and we're going to cycle it back to photosystem 2, and we're going to make it pass through all those proteins again. Why would we do that? Well, the more time those electrons get passed over and over again, the more ATP or energy we are generating to pass to the Calvin cycle. So again, don't forget, the whole point of your light-dependent reactions is to make ATP and to make NADPH. Normally, we make both of them and then we just move on. But if for some reason photosystem 2 isn't working the way that it should and we can't actually make NADP, or NADPH actually, we will just make more ATP. It'll still get used up, it's not going to be a waste, and the Calvin cycle actually uses more ATP than NADPH anyway, so it doesn't hurt the process any. Alright, so here's another visual to help. Again, photophosphorylation. It can happen in two ways. If you follow the blue arrows will show you non-cyclic photophosphorylation, where the ending result is going to be some ATP and some NADPH. The red arrow shows you cyclic photophosphorylation, where the ending result is only ATP. There is no NADPH. Alright, so these are the questions that you should be able to answer at the end of the video. It pretty much goes over all of the high points of what we talked about in this video, and next time you will be looking at photosynthesis as it applies to the Calvin cycle, or the light independent reactions. See you guys in class. Bye.